production for my Tuesday night. Thursday we'll go through this all tomorrow. Yes.
This is for like Thursday lab. Our dichotomous keys are due tomorrow.
For Thursday lab, yes.
At 11:59.
No, at the start of your lab period, so 2:30.
Oh, we got it printed out?
No, it's being submitted through Canvas.
Oh, I'm sorry.
Yeah, you're gonna submit the Canvas electronically, so no, you don't have to hand me anything in the class, but the Canvas submission and assignment page will close at 2:30. So you wanna make sure you've got that submitted before 2:30 tomorrow. In Word format. Okay, all right, excellent. So we were leaving off talking about land plants in our last class, which is essentially what I mean when I say plant. I'm talking about land plants, this specific clade right here, which does have its own shared derived traits that define it, just like we had shared derived traits for the green plants, which included the green algae, in addition to the land plants. This one's just the land plants, they have their own shared derived traits. One of those was the alternation of generations life cycle, which we went into a lot of detail on the board, as well as going through the actual text of the diagrams on the PowerPoint, because I want you to know that one. Backwards, forwards, upside down. Should be able to start anywhere with your sporophyte generation, anywhere on the board, and be able to draw the alternation of generations, whether it's upside down or sideways or anything like that. Know it well enough that you can draw it, because you're gonna have to do that for your test. I'm just gonna tell you now, what you drew on the board, that's what you have to be able to do for your test. And there are two stages in there that are unique to this life cycle that didn't exist in the Haplontic life cycle that we saw for most of the green algae. It was the embryo stage and the sporophyte stage, both of which are multicellular diploid stages. A multicellular diploid stage does not exist in the Haplontic life cycle. It just has a multicellular haploid stage, right? So that's one of their shared derived traits. It's unique to them because it's unique to this life cycle, which all land plants have. Now we're getting into the other shared derived traits for the land plants. Most of these are going to relate to the fact that they're living on land now, right? They're not surrounded by water. They don't have that as a protective means for their cells, for reproduction, things like that. So one of the things that's a challenge when you're dealing with the land environment is desiccation, having your cells and your tissues dry out. One of the ways that land plants deal with this is something called the cuticle. Now the cuticle is a waxy layer that's going to be on all the above ground parts of the plant. It's almost as if you took the top of the plant and you dipped it in wax. It's not actually a living layer. So if you look at the picture right here, you can see these are living cells here, all this portion here, but this little clear layer right here, which represents the cuticle, that's just wax, right? Or just this layer, but it's not made of cells. It's not a living layer. And so what it does is it acts as a sealant, right? A waterproofing layer. So that when the stomata are closed, we'll get to those in a little bit, which are the holes in the leaves, right? When the stomata are closed, the water's not going to leak out as much. There may be some leakage, but it's not going to be that much. It doesn't leak out of the cells or in between the cells and get lost in the atmosphere. If it did do that, then the plant would be in serious trouble of dehydrating and losing too much water to the point that it would actually die. So this is something that's kind of essential for a land plant to have. So all of them have it, including the ones that have gone back to water. So you go to water lilies, water lotuses, and things like that, they still have this. Right? If you live in a drier environment though, something like a desert, you're likely to have a thicker layer of this. So it can vary in terms of thickness, but they all have it because it is a shared derived trait for them. Right? So that's our cuticle. Now, why is it only on the above ground parts of the plant and not the below ground parts? That's where they get the water. You want water to get through the roots, right? You want water to be able to come in. Plus the water's in the soil, so you're less likely to dry out there. So if you had a waterproofing layer on your roots, it really would kind of defeat the purpose of the roots, which is to take up the water. So this is only on the above ground parts that are exposed to the air. All right, the next one we're going to talk about is our first type of cell that I'm really going to discuss in detail. We're going to have a lot of different types of cells that we're going to talk about in this class. There's even one that we won't discuss. That's also part of cells, that's part of plants. But the ones that I'm going to discuss in class are the ones that are actually shared derived traits for different groups or different clades of plants. For the layman plants, the one that is a shared derived trait that for them that's unique to them are parenchyma cells. Now parenchyma cells are a part of what's called the ground tissue. Right? They're cells, so they make up tissue. All cells are going to make up tissue. So each one of these cells that I discussed with you, I'm going to tell you what the name of the cell is, but I'm also going to tell you what the name of the tissue is that they make up. Know what tissue they go with, right? And know which one's the cell versus the tissue. This is the cell, parenchyma. The ground tissue is the tissue, right? And that's most of the plant, right? That's almost all of the plant. If we look at the diagram here, you can see the different types of tissues in plants. There's only 3 of them. You have dermal tissue, you have ground tissue, and you have vascular tissue. The dermal tissue is the darker green right here. That is essentially the thin layer that's around the entire plant. It's essentially the skin, right? That's why it's called dermis, okay? That's gonna be just a thin layer on the outside. And then you have the purple right here. That's vascular tissue, which we will definitely talk about those in detail. We will talk about the specialized cells that go with that. Those are the veins of the plants, the part that's responsible for transporting things. Right? All the rest of it, right? All the light green that you see here is ground tissue. It is most of the plant. Right? And by vast majority, most of the cells that make up that tissue are parenchyma cells. There are a few other types, right? But most of it's going to be parenchyma cells. So you can imagine it's a pretty important cell for plants. Now what I'll do when I introduce cells to you is I'll talk a little bit about their characteristics. So what kind of shape are they? What's going on in terms of their cell wall? And sometimes some of them are going to have what's called a primary cell wall. Some of them are going to have both primary and secondary. Are they alive? Are they dead? Are they semi-alive? And all these kind of characteristics that are important. And I'm definitely going to give you their functions. Okay, all of those are things I want you to know about those cells. So let's talk about the characteristics first for the parenchyma cells. Right? This is kind of your traditional classic looking cell. Right? So the cells we're going to talk about later are elongated, which means they're long and skinny. They look like tubes. Right? But this looks like a cell you would expect to look. Right? It's either kind of rounded or many-sided or kind of cube-shaped, typical shape for a cell. Right? Second thing to know about it. They are only going to have a primary cell wall. They don't. Most of them. There's going to be exceptions, but we're just going to make a generalization here. There are just a few that have a secondary cell wall, but most all of them have just the primary cell wall, and it's fairly thin. Now I'll get into what the 2 types of cell walls are later, because when we actually get into some of the cells that have that secondary cell wall, we need to talk about how the walls form, because they don't form necessarily in the order or the position that you would expect them to. So just right now, just let you know, these have primary cell walls, not secondary. They are also alive at maturity. You might think, well, that's kind of obvious. They're cells. These are living organisms. You'd think that they have living cells. But plants are actually going to have some cells in them that aren't living, or that are semi-alive, and can still function perfectly fine. And we'll talk about what those are later. These, though, are going to be alive and fully functioning cells. They also are what we classify as being unspecialized. Now, unspecialized means that they do not have a special structure or form that allows them to do a special job, right? So like I said, these later cells, we're going to talk about the ones that are tube-shaped, they're dead, they've got really thick secondary cell walls that are waterproofed. They're designed for transporting water. That's all they do. That's their only job, and they are specialized in their structure and their shape, and their form is specialized for that job. These aren't. Right? And that means they're actually going to be able to do a lot of different things for this reason. Right? And one of the very important things that they retain the ability to do is they retain the ability to divide. Right? These other ones that are dead at maturity or semi-alive, they can't do that. Right? They're not going to be able to divide. But these are important because this adds what we call plasticity to the plant. It allows it to respond to stimuli, to a changing environment. If it was rigid and all the cells can only do one thing and are specialized, it's stuck in that form and position. And it's not going to be able to adapt or change or acclimate to new conditions that might be in that environment. So having some cells that have this plasticity, have this ability to be unspecialized and retain the ability to divide and grow is a benefit. This also allows them to respond to injury. If they get hurt, these are the cells that are basically going to repair those wounds. Okay. So these cells do a lot of things. They perform most of the metabolic activities of the plant, and they're very versatile. There's a lot of different things that they do, some of which would seem very unrelated. So, for example, they're the ones that are responsible for the process of respiration, cellular respiration, mitochondria. They are also the ones that are doing photosynthesis. They've got those chloroplasts and they're doing photosynthesis. They can do gas exchange, moving gases around like oxygen, carbon dioxide, things like that. They can do nutrient transport, but I'm gonna be a little more specific here, because usually when we think about transportation of stuff in the plant, we think of vascular tissue, not ground tissue, right? Because the vascular tissue is moving the nutrients up from the roots where the nutrients are in the soil up to the tops of the plants through the specialized cells that are in the xylem, right? So that's kind of your vertical transport, right? Lateral transport is going this way though, right? You've got more than just the veins in the plant. You've got cells that are all surrounding those veins, and they all need nutrients as well. So how do they get it? It has to come out of the xylem at some point and be released into the cells that need it. So you have to have some lateral transport, and the xylem doesn't typically do most of that, right? Getting it into the cells that are around it. That's done by the parenchyma cells. It's responsible for that nutrient transport laterally. And then we've got food and nutrient storage. Some of them will have specialized plastids in them called amyoclasts that actually are responsible for storing the starch. And then we have our regeneration and wound healing. And the only reason they can do this is because they can retain that ability to divide. You can't repair a wound unless you can continue to have cells dividing and create new ones to fill in that gap. So they do a lot of stuff. Very versatile cells, yes.
So are they like the plant version of a stem cell, like a human stem
In a
cell?
way, yes. Yes, because stem cells are unspecialized as well. They haven't decided yet what they're gonna be. But stem cells usually at some point do decide what they're gonna be. Not all of these are gonna do that. Most of these are gonna stay unspecialized. There are a few of them that can turn into a different type of cell. It's called parenchyma, which does serve a specialized function. It's a living cell, but it forms the function of structural support, and that's all it does. But if they stay like this, then they're unspecialized. Yeah. But yeah, it's a good comparison, yes.
What's the difference between how fast the plant heals versus the human?
Oh, that's a good question. I actually don't know the answer to that. I'd have to look that up. And it may be more of a physiology question, which would be more Dr. Ray's expertise, but I'll check with him and see. I mean, I know some of my plants I'm pretty damaging to my plants, so they can heal pretty fast on me, or they die, one of the two. But I'd be curious to know myself. I'll see if I can find out for you. And this is the weird thing about me. I'm a botanist, but the only house plant that I can keep alive is a Christmas fern, or Christmas cactus. That is the only thing that I can keep alive, because it does not require you to water it. Right? You can just leave it for months at a time, and it's still perfectly fine. So I can keep those in the house. But otherwise, yeah, I don't have a whole lot of luck. Now my window right now in my office is doing okay. I've got some African violets there, I've got some succulents, they're doing okay. But every once in a while I'll forget about them and they'll just start going like this on me. So it's you just never know what's gonna happen with my plants. Just put them out in the yard, put them in nature and let nature do its thing, and it'll take care of it for me. So.
Get some resurrection plants and just water them.
I do have some resurrection plants in my office, in my window, so if all else fails, that'll be the one that I'll work with, so yep, that's a good one. Alright, so this is our last two shared drive traits for our land plants, which are gonna both be types of Gamtangia. We have already talked about these, but we talked about them in lab, in relation to the ferns, when we're discussing the ferns and the gametophyte stage. Which was not the dominant stage for ferns, but it is going to be for one of the groups of plants we're going to discuss. All land plants are going to have these Gamtangia that are essentially the specialized organs on the gametophyte that makes the gametes. Right? So sometimes you'll find both types on a single gametophyte, in which case it would be bisexual. Sometimes you have them on separate gametophytes that are unisexual, either male or female. So since we have those two types of gametes, sperm and egg, there are two different types of Gamtangia. You have the female and the male. So the archegonium is the female one, the antheridium is the male one. This is kind of their structure though, right? And the importance of them. Right? Remember, one of the challenges related to making a transition to land is protecting your cells from drying out. And that doesn't just include your own body cells, it includes your offspring as well, your sex cells. You don't want your egg and sperm to dry out because those are your offspring. They're going to die if they do. So this is their means of protecting those. Right? It not only protects them from drying out, though, it also can protect them to a certain extent from mechanical damage, being hit, for example, right, or cut. And the reason it does that is because on each of these types of Gamtangia, there's going to be a layer of cells on the outside that are essentially sterile cells. It's like a little jacket that surrounds it. So you can see it here, there's a sterile jacket right here for the antheridium, there's a sterile jacket right here that's for the archegonium. if you wanna think about it, they're kind of like sacrificial cells, right? If the plant loses it, it loses it, right? They can live without those, they're not that important. Their job is just to protect what's inside of that Gamtangia. What they really wanna protect are these, okay? The egg cell right here, okay? And all of the sperm cells that are being produced inside here. So these can be sacrificed, but they have that layer to protect them, right? Which you didn't need when you were an aquatic organism living in the water. You didn't have to worry about protecting this because it was surrounded by water. Okay. Now the male one is gonna be the antheridium, so that one can only produce sperm. Okay. So if a gametophyte only has antheridia, it is functionally male. That's all it can do is produce sperm. If it is gonna be functionally female, it will have just the archegonia, right, which is the female Gamtangia that makes the egg. If it has both, that's when it would be bisexual. But you won't see a situation where an antheridia is making the eggs as well as sperm, and you won't be a situation where the archegonia can make both sperm and egg. They can only make the one, okay, archegonium, egg, antheridia, sperm. And then as a comparison, what were the specialized structures on the sporophytes called that make the spores? Sporangia, exactly right. We've kind of seen the comparison here, Gamtangia, sporangia, yep. One makes spores, one makes gametes. Okay. One's on a gametophyte, one's on a sporophyte. That will hopefully help with a little bit remembering those. All right. Now what we're gonna do is we're gonna break down the land plants into their different groups and talk about some of the different clades within that, but also some of the paraphyletic groups as well. So we're gonna start with our simplest plants, the ones that we didn't get a chance to do in lab because there's just never time to do these plants in lab. And those are our non-vascular plants. So even though I didn't show them to you in lab, I told you we'd get back to them in lecture, so we'll discuss them now. There we go. Okay. So again, I'm going to try and help you visualize these with the cladograms because we're going to be using cladograms this whole semester. This was the very first cladogram we were looking at that was related to something plant-like. Right? We took our green plants, which included all of our green algae right here, plus our land plants, and made it into that one tree right there. Right? Now what we're doing is we're going specifically to this group because this is the one we're talking about. There's those shared drive traits we've just been discussing right there. We're taking this clade and we are opening it up so we can see all the branches that are inside of that. Okay? And you can see there's several different lineages that are more primitive than the other ones that branch off this direction. That's what we're going to be discussing today. Okay? That whole cladogram is going to be our land plants though. The non-vascular plants are a paraphyletic group within the land plants. Okay? And I've kind of drawn it with a dotted red line so you can see where it's at. It's just these lineages right here. So you know it's a paraphyletic group because if we go down here to this ancestor that's included in this dotted red line, this ancestor right here is also an ancestor of vascular plants, like trichophytes. So anything that relates to these shared drive traits down here that we just discussed has to be for the whole group, right? It's not just for this group right here, right? It's not gonna have any traits that define it. We're gonna keep saying that over and over again. It's a paraphyletic group. They're also sometimes called bryophytes because the biggest lineage within the non-vascular plants are the mosses, and their phylum name is Bryophyta. And so they sometimes call all of them bryophytes. There's three major lineages: liverworts, mosses, and hornworts. And we'll break each of them down into different groups and talk about their characteristics and how you tell them apart. Okay. These are not real complicated plants, right? And I say that in a joking manner because we're starting to realize exactly how complicated plants are, right? But relatively speaking to the other ones, especially what we're working on in lab right now with the flowering plants, these are not that complicated, right, in terms of what they have. Matter of fact, because they're a paraphyletic group and they don't have any shared derived traits that define them, they are actually defined more by what they don't have rather than what they do. Because there is no trait out there that we can say this one is the defining feature of this group. It's found only in them and nobody else. Those don't exist in a paraphyletic group. what don't they have? Well, the first thing that we're gonna talk about that they don't have is something called lignin. Now, lignin is going to be a substance that you find in the more evolved plants that provide some structural support and some stability. It's what makes them hard. So think about a woody plant. That's essentially what the wood is, right? And that is gonna allow those plants that are more evolved to grow taller because they have something, some support in their cells to keep them from collapsing against the force of gravity, right? Or against wind for that matter. Right? But these don't have that. They don't have this lignin in their cells, so that means they have very minimal support, just like a green algae would. And if you recall, when you reached into that water and pulled the green algae out, even though it was nice and feathered out in the water, when you pull it out, it collapses on itself. And that's what would happen with these if they tried to grow too tall. And so they stay pretty short and compact. So here's kind of an example right here. That's pretty typical height, pretty typical size for one of these non-vascular plants. Not real big, okay? Second, they don't have vascular tissue, hence the name non-vascular plants. Hopefully that will help us with the memorization of this one, which means they don't have the veins in the plant to help transport things throughout their body. They are going to be very inefficient at this, okay? That means that they can't really get to be too tall or too big because the only way they can move water and nutrients is from cell to cell to cell to cell through osmosis and diffusion, which is a slow process, right? The veins, they can move it up pretty fast, but if you're going through osmosis and diffusion, you have to go from from one cell to the next and get through all of these cells just to get to the top of the plant. That takes a long time. And if you were too big, you would never be able to move the water and nutrients fast enough to survive. You can't be real big. So that's one reason they're real common in wet and moist environments, and why they're fairly close to the ground. They grow more out than they do up because it keeps most of their cells in contact with the soil. Right? So they have less of a problem with this. That's kind of what aquatic organisms do. They have water surrounding all their cells. This is trying to keep as many of those cells in contact with the water as they can. Now, the irony of it is that these two characteristics in some ways go hand in hand. Okay? In the more evolved plants that have lignin, most of the lignin is actually found in the vascular tissue. So if you don't have vascular tissue, you don't have lignin. If you don't have lignin, you don't have vascular tissue. So you can kind of think of those two characteristics as being paired together. And I'll do that periodically during the semester, is kind of give you some characteristics paired together to help you remember that. Okay. Now, because they don't have vascular tissue, there are two very important features that we normally associate with plants that they're also not going to have. They're both organs on plants, and that is leaves and roots. Right? Because you can only be a true leaf if you have vascular tissue, even if it's just one strand. You can only be a true leaf if you have vascular tissue in you. And the same thing applies to roots. Right? Now, when you look at these non-vascular plants, they are going to have parts of their body that are green and kind of flattened. And some of them even have things that look a little bit like leaves, but they aren't actually called leaves because of this feature, because that's lacking. So they'll be called something like a thallus, or they'll be called something like a phylloid, or something like that. They're not called leaves, they're called something else. But they still do the same function. They do photosynthesis. Same thing applies to our roots, but this one I'm actually going to give you the term for it. Right? They don't have roots, but instead what they have is they have something called rhizoids. Rhizoids function exactly the same way that roots do. They attach the plant to surfaces, hold them in place, anchor them, and they also absorb water and nutrients. Okay? But they don't have vascular tissue in them, so they're not classified as true roots. They're also going to be missing something else called endodermis, which is also a defining feature of roots that they don't have. They are very thin. They're only one cell layer thick, so they look like little hairs. And you can kind of pull the plant back and look underneath it, and you'll be able to see those. As they attach to the surface. Just be a little delicate with them because they are very delicate. But those aren't root, those are rhizoids. Not to be confused with rhizome, which we talked about before as a way of reproducing asexually by producing underground horizontal stems that are not roots, right? That new little plants will sprout up along the nodes. Rhizomes and rhizoids are different things. Rhizomes are stems for asexual reproduction. Rhizoids are the root-like structures that we find in non-vascular plants. Okay. They also don't produce any of the other things that we associate with plants, like pollen, seeds, flowers, fruits. They're going to reproduce with spores. But one thing that they do have that's unique to them, compared to all the other groups, is their dominant stage in the life cycle. They are gonna have the gametophyte, the multicellular haploid stage, as their dominant stage. So when you're looking at the moss, again, the moss right here, the green stuff that you see here, that is the gametophyte stage. That's the multicellular haploid stage. It's free living, it's photosynthetic, it can do everything for itself, it can get its own nutrients, get its own water, make its own sugars. Okay? Whereas the sporophyte, which is gonna be very small, and ephemeral means it doesn't hang around for very long, basically does its job and then it's gonna drop off. Right? The sporophyte is gonna be attached to the gametophyte and nutritionally dependent on it for pretty much everything. There are some types of sporophytes in non-vascular plants that can photosynthesize, but some of them can't at all. In which case, they're gonna have to get all their sugars from the gametophyte below them that they're attached to. They also get their water and their nutrients from that gametophyte. In mosses, that sporophyte tends to look a little bit like a golf club or a little shower head. But we'll get into the morphology of the sporophytes for each of the groups in a second. Okay, first group. First one we're going to talk about, the one that branched off first here, those are going to be our liverworts. Fun fact, a lot of times people named plants after what they looked like. Somebody thought that the growth pattern of a lot of these liverworts looked a bit like a human liver, and so that's why they called them liverworts. People also used to believe back in the day that if a plant was shaped like a body part, it was something you could consume to heal that body part. Right? But they used to think that was the way it worked. Right? But these are named after their shape, their body shape, or at least the ancestral form. There's about 9,000 species of them. It's a relatively small group, actually, considering that orchids, just the orchid family itself has 20,000 species in it. This is all liverworts. And they really like those moist shaded habitats, mainly because they don't have that vascular tissue and they want to keep most of their cells in contact with that wet soil. Now, the fun thing about liverworts is they can look like a couple different things. And generally speaking, we usually group them into two different categories based on their appearance, what they look like, and what those leafy-like structures are shaped like. So we have what are referred to as the thalloid liverworts, which kind of looks like a paint splotch or a drop of paint. And then you have, these quotation marks here, leafy liverworts, which look a little bit like a spike moss, actually. Now the second one down there is thought to be kind of the more evolved of the two. You can see how it compares appearance-wise to more modern-day plants. It looks more like them. So let's talk about the ancestral one first, the thalloid liverworts. So we actually have a term here for their leafy-like structures that they use for photosynthesis. Okay, they refer to it as a thallus, hence the name thalloid liverworts. That's where it comes from, because that mass of green that you see right there, that's called a thallus. Okay, it is a flattened tissue that is functioning for photosynthesis, which means that it needs to be able to do things like gas exchange to get the carbon dioxide that it needs to do photosynthesis and release excess oxygen. And it does that by having pores in the surface of it. But most people, when they think of pores in plants, they think of stomata. But stomata are specialized pores because they are actually pores that the plant can control and open and close, and depending on availability of water. But these pores that we find in the thalloid liverworts can't do that. They are permanently open, okay, which means there's always a risk of losing too much water through them. Because when you have open pores like this, yes, you can get carbon dioxide in that you need, but you're also losing water at the same time through the process of transpiration and evaporation. And so that's also one of the reasons why they like those moist habitats, because they don't have that control of water loss that some of these other more evolved plants do. And then some of them can reproduce asexually. I use something called gemma. And some mosses will do this as well. Gemma are essentially little pieces of plant tissue, okay, that break off and can get either thrown to a new environment or splashed to a new environment through raindrops that will form into new little plants that are genetic clones of the plant they came from. It's an asexual means of reproduction. A lot of times for the liverworts, they'll house them into cups. They house them in little structures on their leafy-like thallus that look like little teacups. Right, so this is what's called a gemma cup. And then the little bits of tissue, the actual gemma themselves, are in the center of that. This is a side view of it right here. So this is the gemma cup. Okay. And then those are the pieces of tissue right there, the gemma. So if a raindrop splashes on that, it will knock out some of these pieces and it will grow into a new plant. Yes?
Is the use of gemma any different from clonal fragmentation?
It's essentially the same thing. It is an example of clonal fragmentation. Clonal fragmentation is more of a generalized term. It applies to any situation where a piece of the plant will break off and form into new individuals. So, for example, if you take an African violet leaf and cut it, put it in water, it'll form roots and it'll make a new plant. That's clonal fragmentation too. When a prickly pear cactus loses one of its digits and it lands on the ground, it's forming into a new plant. That's clonal fragmentation too. So this is a type, but it is a specific type. Kind of like binary fission is a specific type of asexual reproduction. Yes?
What's the success rate for this kind of reproduction?
This is pretty good. Asexual reproduction in general is usually pretty successful because you don't have to find a mate. It's pretty inexpensive, and the risks are less there. But the problem with this is, remember, anytime you have asexual reproduction, you have genetic clones. You have a population that is going to have a little bit of a lacking of diversity, genetic diversity, and that can put the population at risk. Right? Maybe not necessarily the individuals and their ability to reproduce, but it puts the population at risk. They will also reproduce sexually as well. They can do both. Okay, now the leafy liverworts, which look a little bit more like what we think of as plant, they have these things that look like leaves. They're not leaves because they don't have vascular tissue, but they look more like leaves, little scales that you see right here. And they'll have 3 rows of these. Right? They have 2 that kind of go on, split on the side here, and then they have 1 layer that sits right here on top of that. And they do tend to lay prostrate, again, trying to keep those cells in contact with moist soil. So you can see how on this rotting log right here, it's kind of just plastered itself against it because it's nice and moist on that log to kind of keep all those cells moist. It does look very much like a spike moss. But true spike mosses, which are actually vascular plants, they're going to be a bit bigger than these are. A little more defined, and they can tend to grow a little bit more upright than these can. Okay, now what we'll do for each of these lineages is we'll talk specifically about the morphology of the gametophyte or the gametophyte, and then we'll talk about the morphology of the sporophyte. So we've been looking at the gametophyte so far. That thallus that you saw for the thalloid liverworts and the leafy liverworts that you saw, that's their gametophyte. That's their dominant stage. Okay? That means that those structures that you're seeing are going to have the gametophagia on them, the organs that are responsible for making the gametes. Okay? Now, where they're positioned, it does depend a bit, but there are some of them, they're going to form these really unique looking structures right here. They're called antheridiophores and archegoniophores. They look like essentially little palm trees, right? That's what they look like. And we'll get into the specifics of those in just a second. But all non-vascular plants have gametophagia. Where they're located, they'll depend on whether you're talking about a moss, a liverwort, or a hornwort. Not that you can't see it on my slide pictures, but there's something about the wooden models that I just really like. Okay, we're going to look first at an antheridiophore. So this is going to be the male one. This is what the antheridiophore looks like this. It's more flat-topped than the archegoniophore. The archegoniophore has those things that look like fingers that kind of stick down. This is more flat-topped. And you'll see inside of this, I'm gonna kind of bring it a little closer, these rounded oval shaped things right here, those are the antheridia, right? The whole thing is called the antheridia 4. The little individual circles here are the antheridia. So that's where the sperm are actually being produced, right? In the antheridia, on the antheridia 4. I've got two different versions here of the female one, and what's referred to as the archegonia form. And that's because I have one before fertilization has occurred, and I've got one after fertilization has occurred. Okay, so if this is the female one, this is the archegonia form, then it has to have the archegonia on it. That's what these little dangly things are that are sticking down on the bottom of this. Okay, so they're right here. Egg's way up here, but you got this little tunnel that you've got to go through as the sperm. So these are the archegonium. The egg's going to be up here. When the sperm is ready for fertilization, these plants are still going to require a thin film of water for sexual reproduction. They still have sperm that swim. Okay? So the sperm still does not have to swim to the egg here. So if you have a thin film of water here, the sperm can make its way, swim up into this tunnel right here, get to the egg and fertilize it. Okay? But that also means that the sporophyte, which is formed after that process, is going to start developing on this structure. Because the egg is right here, it doesn't move, right? And so therefore, once it gets fertilized by the sperm and makes the zygote, it starts forming into the embryo, which then forms into the sporophyte. So that's what we're gonna see on this one right here. Okay, so looking again at where those little dangly things were, now you see something that looks a bit bigger. It's kind of oval shaped, it has a little stalk on the top of it right there. That's our sporophyte. It's not real big, but it shouldn't be because the sporophyte in non-vascular plants is not the dominant stage. That's about as big as it gets. A close-up of it, this is what it looks like right here. So that's one of those, stuck on that archegonia floor. It's non-photosynthetic. So it can't photosynthesize. It's getting everything that it needs from the gametophyte that's supplying that for it. And it doesn't last very long, just long enough for it to make the spores. That's all it needs to do. Most of it is made up of the sporangia, right, which is called the capsule. So notice this whole thing right here, this whole thing is the sporophyte. Right? But only the round portion down here at the bottom is the actual sporangium. Remember, there's a hierarchy here. Right? You've got your sporophyte, okay, and which has sporangia, which makes spores. Okay? Sporophyte, sporangia, spores. Okay? So we've got a hierarchy going on with that. So if we want to see in the picture right here, this whole thing is the sporophyte. Most of that is the sporangia, right there, and those are the spores. And that sporangia is called the capsule. Okay. All right, now we're going to switch over to our next lineage, which are going to be our hornworts. This is not a big group at all. This has only got 100 species in it, which is one of the reasons why I don't have any representatives that I can show you. I can actually show you a liverwort because you can purchase those. You can find those around here in Big Cypress and Beach Ridge, but hornworts, less likely to find those. They're much rarer. They're usually more tropical, so I don't have any of those. But they have a feature that the liverworts didn't have, and that's stomata. So that means they're going to have pores on them that they can control, whether they're open and closed, to help prevent some water loss. Here's the way that stomata work. They have specialized cells that surround a hole. Okay, so this is the hole right here, the pore. Here are the two specialized cells, they're called guard cells. They're usually long and skinny. They can look like little sausages or they can look like little dumbbells, depending on what species you're talking about. But you can see that there are microfibrils, these stiff fibers, okay, that are kind of form rings all along the distance of the cell that prevent the cell from being able to expand outward. Okay? It can't really get wider this way, but it can get longer. It can get longer, just not wider. Okay? So when there's lots of water available in the plant, water is going to move into those cells, and the cell is going to get longer. Okay? It's going to become turgid and get longer. And you can see the tips here touch each other from one guard cell to the other, and they push against each other, and it actually pops the pore open, okay, in the process. Now, when there's a lack of water, the plant's losing too much, it's kind of ingenious that this is how it works with water, then the cells are going to start losing water. The plant in general is losing water, so the cells are going to lose water. It's going to come out of them, they're going to start to shorten in terms of the length, right, and they're no longer pushing on each other on the tips anymore, and the pore actually collapses on itself. Okay? And it works to their advantage. They don't want the stomata to be open when there's a lack of water, and it closes when there's a lack of water because it is based on water. So it works kind of brilliantly, if you think about it. But these have that, whereas the liverworts didn't. They had pores that were constantly always open. Now the hornwort gametophyte looks really similar to the thalloid liverworts. It's hard to tell them apart sometimes. They both kind of look like paint splotches, okay? Or drops of paint. But here's one difference between them. In the internal space, or the space between the cells, in a thalloid liverwort, there's air. Whereas if you look between the cells, okay, on a hornwort, you're actually going to find a mucilage substance, kind of like, it looks like snot. Okay? This sticky, thick, almost gel-like substance in between those cells, in those cavities. And there's going to be something living in there. Okay? All of these species, all hornworts, actually form symbiotic relationships with nitrogen-fixing cyanobacteria. They live in these cavities, mixed in amongst the mucilage. It's one of the reasons why sometimes these hornworts can look kind of bluish, kind of a bluish green here, because the cyanobacteria has that kind of rich bluish green color to them. So it kind of tints them that coloration, which helps you distinguish them from the liverworts, because the liverworts are going to do this. Okay? So they get that blue-green color from that. But here's why it's an advantage for them. And remember, the only organisms that can fix nitrogen in nature, prokaryotes. That's it. Bacteria. They're the only organisms that can do that. And if you hear about nitrogen-fixing plants, those are plants that form symbiotic relationships with nitrogen-fixing bacteria. The plants are not fixing the nitrogen. The bacteria are. Okay? So these right here are making nitrogen into a form that the plant can use. And they're doing a process that plants can't do themselves. Plants can't convert nitrogen into a form that they need to be able to take up. This organism can. Cyanobacteria can also photosynthesize, which is why this still works. All right. Everybody know what the nitrogen fixation process does? I don't remember. Nitrogen fixation is taking N2, which is the form that nitrogen is in the atmosphere, not a usable form of nitrogen for plants, and converts it into ammonium. So this is ammonium. And plants can take that up, and that's something that they can take up. But only organisms that can do this, prokaryotes. And there's different groups that do that. All right, so we've talked about the gametophyte, and the gametophyte has the gametology on it. They are embedded in the gametic tissue, so they're not going to be in nice little or structures like this that are kind of hanging up above the gametophyte that look like little palm trees. They are actually embedded in the tissue of the gametophyte. And there's layers of cells over top of them that, when fertilization is ready to happen, will actually dissolve, okay? So those that get disintegrated, right, over the surface here, when the sperm is ready to swim over here and actually get into the egg, okay? And when it does get fertilized, the egg gets fertilized, it will start growing a stalk, okay, straight up out of this. That's what's referred to as the sporophyte, which is where these organisms are gonna get their name. It looks like a long skinny horn, and that's why they call them hornworms. All right, we'll stop here for today. It's a good place to stop. And I will see some of you tomorrow. The rest, I'll see you on Monday, and we'll have your test results. Be on the lookout in Canvas for the grades before that, though. Are you throwing stuff on? Throwing stuff on? Yeah.